All right. My name is Vijay. Thank you to the organizers. This has been a very fun meeting. I'm not a mathematician. I'm terrible at math. I am a biologist, and so I'm going to tell you about biology. I'm going to tell you about a problem that we think is quite interesting. I hope maybe some of you think it's interesting, and I'm curious to know what you guys think. The problem that I'm going to talk about is really trying to understand if there's memory inherent to the packaging of DNA into the nucleoprotein complex called chromatin that regulates virtually every essential nuclear function in eukaryotic cells. And Mike gave a really nice introduction of chromatin yesterday. This is like one of the most important. This is the most important slide of my slide deck. The work I'll tell you about, I'll touch on was done by Megan Ostrowski, who's now a PhD student at UCSF, Marty Yang, a former postdoc in the lab, and then it was an international collaboration with Francesca Mattaroli's group at the Hubrecht Institute in Utrecht in the Netherlands. My lab, part of my lab works on technology development. We develop new molecular methods to better understand how chromatin, this nuclear protein complex I just mentioned, is actually structured in cells. And then we really care about chromatin biology. So we apply these new technologies to make basic scientific discoveries in the field of chromatin. In the interest of time, I'm going to talk a little bit about some of the new methods that we've developed. Most of the methods that we develop in the lab really focus on, you've heard a lot from multiple talks over the last couple of days about single cell biology. Our lab develops single molecule tools that allow us to measure the structure of a DNA wrapped into things called nucleosomes at single molecule resolution, either in vitro, in purely reconstituted biochemical reactions, or in fact in different cell types. And one area of chromatin biology that we've become fascinated by, that I'll tell you all about in the next 15 minutes or so, is understanding how chromatin is replicated, right? So if you open up molecular biology of the cell, there'll be an entire chapter, right, dedicated to DNA replication, right? So inheritance of propagation of genetic information is entirely dependent on the fidelity of the replisome, the DNA replication machinery. And the replisome synthesizes DNA, right, semi-conservatively, in an asymmetric process. It creates a leading strand and a lagging strand, right? And so this inherently asymmetric process has to, in eukarya, contend with the packaging of DNA into chromatin. And that's kind of shown in this cartoon here. So in this case, you can imagine a replication fork. This is a traveling molecular machine that will replicate DNA. And it has to plow through a series of nucleosomes. This is about 150 bases of DNA wrapped around an octameric complement of histone proteins. And again, Mike talked about this yesterday. And it has to do so in a way that preserves information. So chromatin is thought to carry some amount of information. It's generally thought that where nucleosomes sit will be keeping the underlying DNA inaccessible. And so things like transcription factors, repair machinery, RNA polymerase, right, cannot access the genetic template, right? So there are a suite of macromolecular machines that serve to reposition nucleosomes, change chromatin accessibility to enforce cell type specific nuclear programs, right? And so when the replisome plows through chromatin, it has to create two daughter strands, but then it also has to create chromatin and assemble chromatin on that newly synthesized DNA. Now, if the replisome was simply recycling the nucleosomes that were in front of it onto the newly synthesized DNA, there would be a problem. Basically, the dosage of chromatin would halve every cell cycle. And that's not what happens, right? What ends up happening is that the dosage of chromatin is normalized by a newly synthesized histones. So there's a pool of nucleosomes that are recycled by the replisome, and then histone chaperones will take newly synthesized histones and deposit those so that both the lagging and leading strands have a similar chromatin dosage, and that chromatin dosage mimics what was present on pre-replicated DNA. Does everyone follow? Feel free to interrupt. Do you have a question?
Nope.
Okay, cool. It turns out that the replisome and the replication fork is an incredibly complex regulatory apparatus. So on the replisome itself, there are surfaces that are chaperones for histones. They grab the nucleosomes in front of the replication fork and through as of yet unknown mechanisms, recycle the histones onto either lagging strands or leading strands. And so there are specific surfaces on the replisome that have a bias towards shuttling nucleosomes to the lagging strand. And then there are specific surfaces on the replisome that have a bias towards shuttling things towards the leading strand. Okay? We were excited because it turns out that the structure of this pretty complex molecular thing, right, it remains undercharacterized, right? We don't really understand how nucleosomes are spaced on newly synthesized lagging and leading strand DNA. We don't really understand how daughter chromatids are asymmetrically assembled. So we know very well that the lagging strand and leading strand synthesis machinery are very different, right? But we don't really know how daughter chromatids are differentially, or whether they are differentially assembled on lagging versus leading strand chromatids, and if there are mechanisms that specifically help resolve structures that might be found on lagging versus leading strands. And then there's also this problem of like position memory. So I mentioned earlier that where nucleosomes sit is quite important. It's not just where they sit, it's also how they're modified, but I'm not going to really talk about that. And so there's this question: does the replisome know, for a given position along the genome, know to recycle a nucleosome that was positioned in position 1 back to position 1 on both lagging and leading strand DNA, right? The same question can be asked about chaperones that are depositing the newly synthesized histones as well, right? And if there isn't positional memory, how does the cell actually reform the structure that was present on pre-replicated chromatin? These are questions that we care about. And one could imagine thinking quite mathematically about how these things happen, right? And going all the way from biochemistry to maybe like a model of how this works and what the potential functional consequences might be. So we name all of our methods after like my favorite Indian foods. And so we published this paper last year, and this is a method that we invented. You know what RASM is, right? It's a backronym for replication-aware single-molecule accessibility mapping. And so we developed a single-molecule method that allows us to maybe get at some of these questions. The way it works is that we take cells, we feed those cells a nucleotide analog called BRDU. The cells will metabolize that BRDU and then incorporate that into replicated DNA, right? We then take the cells, we purify nuclei from those cells, and we expose the nuclei to a highly active methyltransferase called EchoG2. And this methyltransferase is an enzyme that will mark accessible DNA, so stretches of DNA that aren't protected by protein-DNA interactions, and will mark it irreversibly with a methyl mark on the base adenine. It's an adenine methyltransferase. We then take this DNA and sequence it on a very special single molecule sequencer that, with some help from some computational tools that my lab and others have developed, allows us to natively detect whether the molecule we're sequencing has BRDU incorporated, so whether it's replicated or not, and allows us to detect these methyl marks. So that gives us a single molecule map of where proteins are interacting with DNA. This is a family of assays that my lab and others have been working on for the better part of the last 8 years or so. And this allows us to map chromatin fiber structure right behind the replication fork. And one of the things, so this is we have a model that allows us to basically detect BRDU in human cells or in mouse cells using the PacBio sequencer, and this works in human and mouse cells. And one of the things that we wanted to ask was, how are these newly replicated chromatin fibers actually structured compared to the unreplicated or the steady state fibers? And so we did a very, very simple computational analysis. we computed so in all of our data that I'll show you a little bit in a few slides, the purple is accessible, the blue is inaccessible. That's just the color scheme we use. We compute the per molecule accessibility for the BRDU positive molecules, and we compare that to the steady state unlabeled molecules. And we discovered something that we thought was quite cool, right? It turns out that newly replicated chromatin fibers are hyper accessible. They're way more accessible, 15 to 40% more accessible per molecule to this methyl transferase compared to unreplicated fibers. And this is true in human cells and in
For how long?
Yeah, so I'll get to that, right? So it's true in human cells and in mouse cells. And this period of hyperaccessibility lasts for hours. It lasts for a very long time. It takes most of S phase to actually get back to the kind of steady state that we're measuring. And so and this is and so we were kind of curious, like, what was actually going on here? Is it that on average these pieces of DNA have fewer nucleosomes, or is it something else, right? And it turns out that newly replicated DNA does not really have fewer nucleosomes per unit length DNA. It's that, and this is a histogram of the sizes of the nucleosomes that we're footprinting, newly replicated DNA has systematically underwrapped nucleosomes. So I mentioned there's about 150 bases of DNA wrapped around 8 histone proteins. Each nucleosome on newly replicated DNA is underwrapped. It's systematically underwrapped, and this leads to, in human cells, about 40% extra accessibility per molecule, and in mouse ES cells, which already harbor hyper accessible chromatin, some 15% more accessible nascent chromatin fibers. And it takes hours for this to resolve. So there's some kind of timing element at play. And in work that I don't have time to go over, we can also show that these patterns resolve differentially on lagging versus leading strands as well. So there seems to be some asymmetry at the level of each daughter chromatid as well. And so,
maybe naive question: do the once it's replicated, the marks on the DNA that was there in the unreplicated strand, they transfer over?
So the methylation does.
Yes.
There's
Is the time scale for that along the line of what you found, sort of unwrapped to that time?
It actually is, yeah. So the time scale for histone modification transfer and for DNA modification transfer is also, both of those other groups, Anya Groth's group in Copenhagen has shown that those also take quite a long time to actually get back to. They take longer than a cell cycle, which is a little bit strange to think about. Yeah.
How can we interpret the shorter footprint for your BRDU? Is this is basically how are you setting the size for it?
Well, so this is the I'm going through this quite quickly, but this is the size of a nucleosome.
Yeah.
Right.
That's why I want to.
These are also nucleosomes. They're just protecting less DNA.
And so then, are you thinking of this as potentially then you have a lot of motion of those nucleosomes? So what you're actually protecting is more dynamic.
Well, yeah. So one possibility is that the nucleosomes themselves are more dynamic, right? And that could be caused.
It's frustrating. It takes some time to find the
size of
That's right. They could be the nucleosome could be sitting on a suboptimal piece of DNA, and so you're
But if it's optimal, you still have to search for the right things.
Sure. It could also be the linker histone hasn't been placed yet, and so the nucleosome is more dynamic because of that. All possibilities. Yeah.
Is the recovery time an intrinsic property of replication, or does it change with the cell cycle length?
Yeah. Well, so in every cell type we've looked at, we see nascent chromatin hyperaccessibility.
The interesting point is H1 there or not? Is H1 on the?
Yeah, we think it's probably H1. Yeah, I think it's probably the timer here. It might just be linker histone. It could be a combination of linker histone and topology, right? So the amount of time it takes to resolve the topological state of DNA that is in the wake of the replisome too.
Yeah, I was going to ask. I mean, do you know if this depends on topoisomerase action or?
We'd want to do topo degron experiments. They're extremely toxic, so we weren't able to do we weren't able to do those experiments. Yeah. And we wanted to do linker histone perturbation experiments, also difficult because there's too many paralogs of like the multi-copies. So yeah. Any other questions? Yeah.
Question. So I have a question just about the fiber seq, the measurement of the BDRU. Like, how do you know it's accurate? Like, how have you validated that?
We've done mass spec, we've done nucleotide mass spec, we've done we sequence controls, so we create BRDU containing templates using uracil tolerant polymerases, and so it's calibrated in several ways, and we publish this. Yeah.
2 minutes.
Great.
Have you connected this to any biological consequences, like transcription rates being higher or something?
Yeah. Maybe I'll just. These are just questions that like we have, right? So we haven't done this work, but Anya Groth's group has collected some data suggesting that the hyper-accessibility, the unique chromatin state that occurs behind the replication fork can alter transcription factor binding, right? And so this might be important for like, in some cases, for like lineage bifurcation, right, and lineage commitment, right, where there's this privileged privileged time where specific transcription factors can access, say, enhancers versus not access them, right? Yeah.
So I just have a clarifying question. The footprint size or the distribution that it's being inferred from the methylation data.
We actually infer accessible. We have a 2-state HMM, so we infer accessible versus inaccessible states. So these are the lengths of the inaccessible tracks
And
per molecule.
You have more information than just the distribution of size. You also have the places where the.
That's right.
Yeah.
That's right. And again, I'm not going into that because of time, but that's all in our paper. Yeah.
Can you partition the fast recovery versus slow recovery regions to be able to identify if there's specific transcription factors that are associated with fast or slow recovery?
Yeah, that would be really, really interesting to do. We've certainly looked at like promoters and enhancers, right? And the dynamics of nucleosome recycling are just different there, so that you can classify them into different states. Again, it's something we like went over in the paper, but like promoters behave very differently than enhancers, behave very differently than random stretches of chromatin. But this hyper accessibility is present genome wide. It's not like specific to early versus late replicating regions.
Okay, so maybe in the interest of time, I suggest that we move this discussion to the coffee. But one last question, Vijay. Sorry.
I still have 2 more minutes.
I still have. Okay, we still have 2 minutes, so I'm going to ask a question now. What will it take for you to name the next thing Sambar?
We already had Sambar's coming out soon. Yeah.
Okay, sorry, we can have one more question. I looked at that time, which is not the right time, I think.
Yeah, there's still 2 more minutes. So.
1, 2. Okay.
So I have a question about the replication factory. When the DNA replicates, it is not always in a single strand. A few strands come in one place and then start to replicate at the same time. So I was wondering if you have seen these hyperaccessible regions are not being randomly distributed throughout the genome, but actually forming these hyperstructures.
We see this genome wide, right? So if the question is like, are there like coordinated forks that are structured? I'm sure we could find examples of that, right? It's not something we've really looked at. Is that what you were asking? Yeah. So yeah, no, we haven't looked at that. It was an interesting idea.
So there was all this work like 15 years ago about thermodynamic codes. John Whittam was doing. Do you see any of this sequence dependent or all that stuff that
we love? I mean, work that was done, very close by, right? And so in our paper, we tested the hypothesis that nascent nucleosomes are subject to more sequence-dependent positioning than steady-state nucleosomes because and Jonathan Whittam and Kevin Struhl, many groups have shown that sequence-directed nucleosome positioning is very quickly overwritten by ATP-dependent chromatin remodeling enzymes that slide nucleosomes. And so we actually show that there's a measurable increase in sequence-directed nucleosome positioning right behind the fork, which is quite cool. Yeah.
Okay, one last question there.
Is there any evidence that during these times the transcription factors have more cooperativity, or it will help increase the cooperativity of nucleosomes since the cooperativity of transcription factors since the nucleosomes are dynamic? Is there
yeah, I mean, even measuring like transcription factor co-binding is non-trivial, right? Like bona fide, the evidence that there is pervasive transcription factor cooperativity, right? I think it probably exists, but it's very difficult to measure, right? So measuring that in this context is very hard, right? It is an exciting. One model we would love to test is that these unique nucleosomes are, they're landing pads for specific types of chromatin remodeling enzymes or specific types of transcription factors, right? But we haven't tested that. But it's an interesting idea, yeah.
Okay, let's continue this question. Let's all thank Vijay again for the